Hi, welcome to Pikai Pharmacy. So today we're going to learn about pulmonary drug delivery. Now, pulmonary drug delivery is an innovative approach to administering therapeutics directly to the lungs, offering local and systemic treatment options. This method has gained prominence due to its ability to bypass some of the limitations associated with traditional routes of administration, such as oral or intravenous delivery. Now, as we already know that the primary task of the respiratory system is exchange of gases between the alveoli and the blood. So as we know that there are tiny millions of alveoli within the lungs, and the alveolar region is composed of mainly two types of cells. One is the type 1 pneumocytes and another is the type 2 pneumocytes. The type 1 pneumocytes are very thin cells which covers almost 95% of the total alveolar region, whereas the type 2 pneumocytes are responsible for the secretion of pulmonary surfactants. Specifically, we call them the Clara cells. Now, these surfactants released by these Clara cells mainly decreases the surface tension in the alveoli in order to diminish the effort of breathing and preventing the alveoli from collapsing during expiration. And immediately below the surfactant layer lies a thick lining of fluid through which the drug must diffuse in order to reach the epithelium for the pulmonary drug delivery. Now let's focus on the mechanism of pulmonary drug delivery. The lung actually provides a vast surface area, approximately from 70 to 140 meters square in adults, and a highly permeable membrane which facilitates the absorption of drugs into the bloodstream. The alveolar epithelium is the primary site for drug absorption, making it particularly effective for delivering small and larger macromolecules like proteins and peptides. Now, if we look down towards the lower region of bronchi, the air velocity over there is relatively low. So, the particles deposit under the force of gravity by the process of sedimentation. And the particles which experiences this process are within the size range of 2 to 10 micrometer. And then if we go more down the respiratory region towards the alveoli, there relatively negligible airflow is present. So over there only smaller particles can reach, particles within the size range of 0.5 to 2 micrometer. The particles over there experiences Brownian motion. Thus, the particles collide with the airway walls, resulting in diffusion. So due to this, smaller particles retain longer in the lungs than larger particles. But however, smaller particles also experiences phagocytosis by the alveolar macrophages. So the deposition of particles within the respiratory region occurs by three main processes. One is the impaction, which takes place within the upper respiratory region, and it is faced by particles having size more than 10 micrometer. Then comes the sedimentation, as we have discussed, and lastly the diffusion. Now if we summarize the mechanism of pulmonary drug delivery, so it happens in three main steps. First is the inhalation, followed by the dissolution of the particles. Since dissolution of drug particles is very slow due to the presence of mucus layer all over the respiratory region. After dissolution comes the absorption. So there are some noteworthy things within this process, that is, for hydrophilic drugs the transport occurs through intercellular junctions, and for lipophilic drugs it happens via transcellular transport. Now there are some key advantages of pulmonary drug delivery, which includes direct targeting, that is, drugs can be delivered directly to the site of action in the lungs, enhancing their therapeutic efficacy for respiratory diseases. Then comes the rapid absorption, where drug administered via the pulmonary route can enter systemic circulation more quickly than those taken orally. Thus, it can avoid the first pass metabolism. And lastly, the minimized side effect. So by targeting the drugs to the lung, the systemic side effects can be reduced compared to other routes of administration. Now when it comes to the formulations, so there are various formulations that can be utilized in the pulmonary drug delivery, which includes nanoparticles, since they are designed for targeted delivery and can significantly reduce the required dosage while minimizing the side effects. Then comes microparticles, liposomes for sustained release formulations, and lastly most common dry powder inhalers. as these devices deliver powdered medication without the use of propellants, making them patient-friendly and stable. Now, if we look at the delivery devices, there are three main types of devices that are used most commonly. The first is the nebulizer, which converts liquid medication into aerosolized particles. Now, nebulizers are mainly of two types. One is the jet nebulizer and another one is the ultrasonic nebulizers. So, jet nebulizers operate by the principle of passing air at high speed over end of a capillary tube where the liquids are drawn up the tube from a reservoir following Bernoulli effect. Then comes the ultrasonic nebulizers, where the principle of operation is that it uses piezoelectric transducers to induce waves in a reservoir of solution. Then comes the pressurized meter dose inhalers. So these pressurized meter dose inhalers are very specific in delivering the amount of medication in aerosolized form. So these pMDIs are mostly used to deliver suspension aerosols. And then comes the dry powder inhalers, DPIs, which require patient to inhale deeply to disperse the powdered medication into the lungs. So this DPIs operates by the principle where a capsule is placed in a spinhaler device. So there happens to be a capsule filled with powdered medication which gets pierced by a special mechanism. So this spinhaler rotates the capsule under influence of patient's breathing, which ejects the aerosolized particle into airstream. This particle then passes through a rotor blade and are collected or deaggregated to ensure that only small particles are administered to the patient. Now there are multiple challenges with pulmonary drug delivery. So the first being the biological barriers. So under biological barriers, the first one is airway defense mechanism, where the lungs are equipped with cilia and mucus that traps and expels foreign particles, making it difficult for drugs to reach the target site within the lungs. So there happens to be hair-like structures called cilia over the epithelial cell within the inner side of the bronchus, which propels out foreign particles. It not only propels out unwanted foreign particles, but also it propels out the drugs that we administer. Then comes the oropharyngeal deposition. So a significant portion of inhaled drugs can deposit in the oropharynx rather than the lungs, leading to reduced therapeutic efficacy. Now studies have indicated that only about 20% of inhaled drugs reaches the lungs due to such loss. Then comes lastly the cough reflex. So the cough reflex can expel inhaled aerosols before they even reach the pulmonary target, which further reduces the delivered dose. Now apart from biological barriers, the barriers are also provided by the particle size and distribution. So the size of aerosolized particle is critical for effective lung deposition. The first is the optimal particle size. So particle must be typically ranging from 1 to 5 micrometer to effectively penetrate deeper into the alveolar region. Larger particles are likely to be trapped in the upper airway, while the smaller particles may get exhaled. Now there is another thing that due to the hydrophilic nature of lung epithelial lining, it is very easy for penetration and absorption of hydrophilic substances. So it is easier for hydrophilic drugs to penetrate the epithelial lining. Adding up with the particle size, then there is also aerosol dynamics. So the momentum and flow pattern during inhalation can lead to particle impaction on airway surfaces, which further complicates achieving the desired distribution within the lungs. Now patient compliance and technique also poses a barrier, as effective pulmonary delivery relies heavily on patient adherence and proper inhalation technique. Then comes the formulation challenges. So developing formulation suitable for pulmonary delivery involves several complexities. So first being the drug stability and irritation. So some drugs may cause irritation when delivered directly to the lung tissues. Additionally, formulating drugs that can remain stable in aerosol form while effectively penetrating the lung tissues is very challenging. Then comes the pharmacokinetic variability as a barrier. So the pharmacokinetics of a drug can vary significantly between individuals, especially among elderly patients or those with compromised lung functions, complicating dosage predictions. So when it comes to the formulation, another thing to be noted is that the solid state characteristics of the formulation also matters, as thermodynamically stable amorphous particles tend to be more soluble than the higher order crystalline counterparts. So stable amorphous particles makes it easy for pulmonary drug delivery. And then lastly, one of the main barrier is the disease specific challenges. So in patients with respiratory diseases such as asthma or COPD, additional challenges arises like airway obstruction and mucus production, variability in lung function. So addressing these challenges is crucial for optimizing pulmonary drug delivery systems and improving the therapeutic outcome for patients with respiratory conditions. I know I've been irregular on this channel, so I'm putting out a video after a very while. So this is a video after a long time. I hope you like this video. Thanks for watching. Stay healthy. Bye.
Seems like every time I run, get my legs broke. Falling through the ground where they have fun on the same floor. Have you ever seen somebody's love?